much. Yeah, so there's kind of a diaspora of different biological application areas, even within RNA biology, but then certainly I'm talking about much more generally than that. The cursors. Okay. Very computationally intensive, which means, time, people power. And this is because it is foundationally dependent on references. It's missing biology, and it is also, and I will not have time to talk about this today, I will argue that today's paradigm does not present our data in biology as AI ready for the transformations that we have seen across society in other domains. And that would be a separate talk that I'm not going to give today. So what is this paradigm I'm talking about? It's a paradigm of alignment and assembly. So I'm going to kind of not discuss assembly today. Let's assume that we have an assembled reference genome. What is the first step in essentially every workflow? It is to take that reference genome, take the rich raw data that we gather from any biological sample called reads, and match it. And that matching is very interesting. It was developed by computer scientists, but at the same time, it is not fully mathematical, and it misses some important information and prevents us from as precise downstream analysis as possible. So raw data is reduced to positions. In addition, which misses very important signal. In addition, during this process, it is an algorithm. So it necessarily distorts the raw data and introduces bias. Okay. Okay, so it introduces bias. Blind spots when reads are absent from the reference are missed. Not included. And then this problem persists through other tasks that we have asked aligners to do, such as gapped alignments to discover RNA splicing, mismatch reporting for discovering RNA editing or mutations. And each of these tasks, when aligners are asked to do them, they either struggle or fail in many cases. Okay. For further motivation, I said, assume we have a reference genome. In many human diseases, especially tumors, it went back. We know that is simply not the case. The hallmark of tumor genomes is almost always a complex or not super complex, but a highly distorted genome with respect to the human reference. And yet, the first step in almost every cancer genomics workflow is to take this rich raw data and align it to the human reference genome. That will not capture the complex diversity of the human tumors. And then just for a second, think about other cases than humans. Human genomes have been scrutinized more than any other genome on Earth, billions of dollars poured into them, and yet we only have a few almost complete reference genomes. Even T to T is not fully complete. Again, esoteric side note. In other non-model organisms or complex symbiotic relationships between microorganisms and their hosts, we will not have a single reference genome. All of this is motivation to get away from it if we could. Okay, so even if you accepted the idea that an alignment and assembly and alignment was a perfect computational procedure, there are still downsides to today's paradigm because it is based on what I'll call custom bioinformatics. After alignment, you as a researcher have to choose a single workflow. Like, I want to discover regulated splicing. Another person might want to discover regulated RNA editing. And that's within the narrow realm of RNA. Move away to single cell attack-seq. Many other tasks in human genomics we're all aware of all require their own workflows and specialized algorithms. And those algorithms do not have any generality in terms of like an analytic framework to help us like sort of borrow the knowledge that you may learn an algorithm designed for mutation detection to use that knowledge or inform algorithms for RNA splicing, for example. Okay, so they don't generalize, they're very time consuming, and they lack fully statistical controls. As I said, they're also not enabling us to be AI ready. Okay, so what is SPLASH? SPLASH is a totally different approach to this problem, and let's kind of think about a thought experiment. What if we could analyze raw data without pre-specifying the features we're interested in and find the most strongest signals of regulation? Okay, so as a thought experiment, if we could design an algorithm like this, we would have to find a unifying feature of many of these zoo animals of tasks. And I don't want to, I'm just being sort of silly, like they're extremely important tasks, but splicing, editing, and so on. There have to be a unifying feature of what they look like from an algorithmic perspective. And this algorithm, if it worked, would have to perform the tasks that are traditional today in human genomics, such as splicing, mutation calling, beyond that, antibody repertoire calling, and more and more. It would also work for totally other kinds of problems, such as understanding microbiome, discovering new mechanisms, such as CRISPR or other approaches. So I'm going to tell you what SPLASH is. SPLASH is essentially saying there is a fundamental unit that we can detect of regulation that we can detect in raw data. We don't need to know anything about where those samples came from, as in the reads that generated them. And we don't need to know anything about the biological origin or any ontology at all, actually. So no reference genome, nothing about what a gene is. So we're going to try to detect these signals through statistics, and it works, as I will not have time to tell you, in many different technologies, okay? It should also be able to discover known and novel sequences. So this is what SPLASH is. It's actually quite simple. So today's paradigm is alignment first or interpretation first, and then statistics second. So first you do alignment, then you might find splicing calls, and then you do a statistical test. Are the statistical features of these splicing different in the two different scenarios that I'm testing, for example? SPLASH is simply flipping that order. It is doing statistics first, interpretation second. And that unlocks many advantages. So what do I mean by statistics first? You take any set of sequencing reads that you have. Each of them is depicted as one of these file stacks. What the algorithm does is it essentially searches through all of the k-mers in every file. And then it fixes a single k-mer. Let's just, for sake of discussion, pick a blue k-mer. Blue is some sequence like AT, GGG, etc., about 27 long. The algorithm will do this in parallel for every single 27-mer. But for the blue one, it looks for it in every file. Then it looks right next door to it, which, okay, let's say, or it looks a fixed distance away from it. For the sake of this discussion, it will be 1. And it catalogs what that next k-mer is. In this case, it's, there are 3 different ones in different files. In these 2 files, every time you see blue, green comes next. So a different sequence like GGGGG. In this file, every time you see blue, purple comes next. And purple is a different color than blue. So it's a different sequence. And in this file, red. So what is SPLASH is doing this counting and then feeding those counts of different colors into a statistical test, too esoteric for the time I have here. And it is looking for a signature, very intuitive signature. Once I see blue, if I'm in one kind of file, am I much more likely to see red than if I'm in a different file? So it's essentially a test of independence. It's essentially a test of, once I see blue, does the next color depend on which file I'm in? So after this step, which is a statistical one, we then proceed to the following steps, okay? So which is an interpretation step. I'm trying to go fast, sorry about that. So let's try to give a tiny bit of intuition for why this works. Suppose you have 2 different splicing events, exon 1, exon 2, or exon 1 and 3. Exon 1 is going to be colored blue. It's that fixed K-mer that I was talking about in the past slide. So what if there are two, we don't need to know anything about the gene, anything about the splicing event. What if there are these two variants in our sequencing reads, and they have differential expression at in two different cell types, for example, okay? So in order for Splash to detect this, there's only two ingredients needed. One, there's two different variants. They could be splice variants, could be the same splicing event, but a mutation or allele-specific splicing. And the other ingredient is simply that these two variants are not equally distributed like all the time. Whenever you observe it, whenever you observe one variant, you can totally predict the other. That isn't the case. So there's some regulation. Okay, so whenever you have this, then Splash will essentially develop a test on how, if it develops a test for whether the ratio of orange to purple is different in different samples. And this hints in an algorithm that I'm going to skip over for time that allows us essentially to detect myriad forms of genetic regulation, including at the DNA and RNA level, from mutations to splicing to VDJ recombination and more. Okay, so essentially, when I've talked about what Splash is, sample, so this regulation I've said is, what it means is that the relative amount of different variants, let's say if you want to think about it, the splice variant, those relative amounts are different in different samples. That sample is a very general term. So if you're doing spatial transcriptomics, sample can be a spot on an array, or it can be a barcode in a 10x experiment, or it could be a bulk RNA-seq condition, and more. Sequence is also general. It could be DNA, it could be RNA, it could be methylation data, and you could pick your type of data, it doesn't matter. So what happens is we do statistical inference. Not sure if you can see my cursor. I don't think you can. So we do statistical inference. Then we have an optional statistical assembly that extends. You might be thinking to yourself, 27 is very short. How could you do anything with this? Blue sequence, or even the concatenation of 54, we have a statistical assembly approach that was developed by a team led by Sebastian Durek's group and others. This whole implementation of Splash, I should say, is due to them. And after that point, there are many different options for interpretation, including ones that use deep learning, but I'm not going to talk about. But here, in silico translation directly of the sequences called by Splash, so absolutely no genome assembly or alignment, or you could take the sequences that are called by Splash and then feed them into an aligner for interpretation. So you can also view Splash as simply an ultra-efficient statistical preprocessor, where you would take your raw reads, run Splash, and essentially switch out Splash's calls instead of the raw reads, and then do whatever task in custom bioinformatics you were doing today. Instead, you do it on these reads, okay? It's a very, very fast algorithm, very simple to use. Again, I'm going to skip it. So I'm going to just give you a handful of vignettes. Some for how Splash can detect circular RNA, another for splicing, and another for RNA editing. And again, this is the same algorithm run with no metadata. And by that, I mean no knowledge of any genome, no knowledge of any sample type, no knowledge of the event we're trying to detect until the end. So until we say, okay, here's some K-mers, were there circRNAs in there? Okay, so hallmarks of circular RNAs, very near and dear to my heart. They generally are involved with other splicing events, and they appear to have regulation if you, even in a single biological context, when you treat with an enzyme called RNA-seq. RNA-seq is an exoribonuclease that digests linear RNA. So if you have two samples of RNA, one just not treated, and the other one is treated with this enzyme, the enzyme will reduce the total numbers of linear RNAs but not touch the circles. So what I'll show you next is we kind of blindly benchmarked this approach SPLASH on some data that was generated with and without RNA-seq. So you took normal samples and then also samples with RNA-seq. The y-axis is the RNA-seq minus sample, the x is RNA-seq plus. So in the RNA-seq plus, you expect less linear RNA, more circular RNA. In the RNA-seq minus, you wouldn't expect anything a priori. What we find is that events called by SPLASH, they're enriched already in whether there's the circle to linear isoform of the circular RNA. And then when you, so again, blue is called by SPLASH, red is not. With RNA-seq treatment, that signal increases, as in the blue is shifted left. There's even more circle, which is what you would expect if you were detecting real circular RNAs. And then if you look more granularly, this feature is even more true, that most of the calls by, or the negatives by SPLASH are ones that have very low values on the x-axis, so where the linear RNA is very high. Another example of what SPLASH can do readily is discover varieties of complex events in cancer. So I'm just going to give you a quick snapshot here. Again, we ran SPLASH blindly on all the cancer cell line encyclopedia and looked for some of the events that it called. Notably, it called a gene fusion BCR-ABL, which is one of the kind of hallmarks of this cell line, and it found novel splicing in known oncogenes, including PTEN, along with, so this cryptic splicing, along with mutations that were embedded in the alignment, that it's a clear reason to us why past approaches would not have found this, because spliced alignment or gapped alignment in contexts like this, where the reference genome is actually not accurately representing the real transcriptome, are difficult to call. So, okay, last vignette. We ran on thousands of cells from the Tabula Sapiens Consortium, which are single cell compendium of multiple primary tissues, multiple donors, multiple tissues within each donor. No reads, no genome, and no knowledge of the tissue or the cell type was used. So it's totally blinded data. In this case, this was actually analysis done several years ago now. So reads were subsampled because we implemented the first, the first implementation of SPLASH was amazing, but it was in Python. And so now today, thanks to Sebastian's group, it's ultra efficient. We had to subsample at that time. So one of the anecdotes I want to tell you about is something I think is actually quite exciting and significant itself, but again, just one outcome. And that is the kind of for free running SPLASH enables us to find single cell regulated RNA editing, including hyperediting, which is today very difficult, if not impossible, to find without highly customized and highly computationally intensive procedures. So what do I mean by, so why would you expect this? Well, if different cells are, there's a transcript that is getting hyper edited and different cells have different levels of that editing, first of all, SPLASH would find it. It would fall into this formulation that I had to skip some of the details for time, but the idea that once I see a K-mer, in this case, the blue K-mer right abutting the red, that blue thing is going to be fixed, but the red sequence is going to change it by how much editing has occurred. Okay, so SPLASH should find it, and indeed it does. This is also a classic case where alignment is going to struggle. If you don't know a priori where hyperediting or editing occurs, it is going to be very, very difficult, if not impossible, to precisely quantify and detect all of these variants, especially at single cell level, where I'm not aware that it has been done. So last anecdote, I told you we ran the algorithm on many different tissues. So what we did, we kind of decided to test whether it was computationally finding interesting biology. We aligned the anchor targets to the human genome for interpretation after SPLASH was run. And we then cataloged the genes. For each tissue, we then get a list of genes that have evidence of alternative splicing. So when we look at the eye in donor 3, we get a list. When we look at the blood in donor 5, we get a list. So we get these lists. There are about 18 of them, lists of genes. We then intersect them. So on the x-axis here is the number of tissues where a gene was detected. On the y is the number of splicing genes. We looked at what's going on at this very tail. What are the genes where in every single tissue, the gene is called as having alternative splicing? What was very surprising and exciting was, well, these genes I'm accustomed to in workflows I've even designed and worked on, honestly, where the genes that would be on this list would include housekeeping genes like GAPDH, actin, ribosomal protein genes. Very interesting. There were no such genes here. Instead, there were just 9 genes, 5 of them splicing factors highlighted in orange, 3 involved in core transcriptional regulation of histones. And then the last remaining gene is one of the it's a heat shock regulating protein that is thought to be kind of at the crux of many different signaling pathways. All of these have ultra-conserved intronic regions or highly conserved intronic regions, suggesting that SPLASH was finding new splice variants, or at least detecting ones that we could assign to specific single-cell regulated expression programs. If we relax that, we sort of look at a little bit further towards we don't require as many tissues to be called in the gene list. Then we look at the Go terms. All of the Go terms are involved in RNA processing. Again, none of these background Go terms that I've been accustomed to. For time reason, I'm going to skip, except to say that SPLASH can also find new splicing in very well-studied genes, including microexons, in this case, microexon in a ribosomal protein gene. Okay, so just to summarize and conclude, there are now many different papers that we have published in many domains of life, essentially almost to the point where you can name your domain. So virus, bacteria, plant, human, non-model organisms. We've been able to find what I think are very exciting results from this approach, and I think it is very easy to use, although it's not what is standard today. I also think that it's very exciting to think about and work on how SPLASH can really open the door to new kinds of deep learning approaches in biology, which is an area that we are actively working on. So in summary, there are lots of different applications to different emerging technologies from 10x, from other spatial transcriptomics. And I'm always excited to talk about them and thank the team and funding.
Thank you, Julia, for a great talk and also for shortening due to technical difficulties. I'm going to ask if there's one burning question, and we'll save all the rest of the questions for going up to Julia after the session or the morning session. So anything incredibly burning? I saw your hand first, so get the one question.
Okay, thank you for this opportunity. Yes, I would like to understand how SPLASH handles repeats, because we always face problems with ambiguous mappings. So how do you distinguish? Yeah.
I think that's one of the most exciting directions for SPLASH, actually, and one that I had been working on myself for a while before SPLASH. So when you're asking about handling repeats, so I think one of the could you just tell me which repeat type you want to discuss? So for example, small, like USNRNAs, I'll just give that example. Spliceosomal RNAs, and ribosomal RNAs, they're high copy number type RNAs that you could consider them repeats. I'm not sure if you mean that or if you mean centromeres, but it's been a challenge to be able to determine if there is cell type or whatever regulated expression of repeat variants, because you cannot first map them, as you mentioned. And yes, we did. One of the examples that fell out of this analysis for primary human tissues was variants in spliceosomal RNAs in the U5, I believe, category, where there's strong evidence that there's, cell specific expression of different variants, including expression of annotated pseudogenes. And so another problem today is pseudogenes in these repeats it's hard to really know if their pseudogenes are actually expressed. So it's just a more direct approach. Thank you for the question.